All right, let me get start. So this is week number 8. We are going to discuss 3 topics. First one is support vector machine. So this is a very important classification method. It's a big framework. And then I will mention kernel method, feature mapping function. So as an introductory lecture for machine learning, I have to mention this to make the course complete. Because when machine learning people use SVM, use kernel, they won't explain to you what they are doing. It's just a huge framework. If you have no idea what it is, then you got lost in the middle of the discussion. You have no ability to penetrate understanding the meaning of some of the methodology they developed. We have limited application in single cell data analysis field because, again, as a classification problem is supervised learning method. So in single cell data analysis, we don't have enough high quality training data. So we don't know if it's true or not. You can use as a training data set to optimize the parameters of your machine, your classifiers. Okay. So the purpose of this lecture is to make this whole course complete. But the next topic is really relevant to what we are doing right now, cell-cell interaction analysis. So I will explain to you the principle. What's the idea to use single cell data to look at cell interaction between different cell types? Then I will show you how to use data-driven method to infer that there are some kind of interaction between one cell type to another. Okay, with a direction. So one cell type interact with another, and or that cell type will feedback with communications. Not too hard, but you have to know the principle. You know the software packages, and they claim they can detect that. What that means? Okay. So you have seen this many times. We have dot product between 2 vectors, and their geometric interpretation of this dot matrix, dot vector multiplication. When you manipulate it, U with V, that indicates the norm, the length of this vector, multiplied the length of that, times the cosine and theta between these 2 vectors. So this is a numeric relationship between this and that. And then it's expanded from application of this dot product of the 2 vectors. You can use it to estimate the angle between 2 vectors, because you know the cosine is this divided by that. Okay, so I'm pretty sure you have already done the homework, and then you explain the concept yourself, so you should know this by now. For PCA, and we use the application, the property of this dot product is projection of one vector onto another, so to get the length of this projection. Today, I'm going to show you this relationship. We can derive this distance from one data point to a line. Okay, so you can define a line, which is green line, use a vector d, right? So the distance between from this data point to this line is, because you know this, you know the norm, so it's very easy to compute this distance. And the change the coordinates. That's exactly what PCA is doing. Okay. So last week we discussed the difference between PCA and LDA. This serves different purpose. PCA is purely rotation of the data points in order to show the diversity, the variability among data points. Well, LDA serves different purposes, try to rotate the data point as well. But these data points are labeled as red and blue. The purpose of rotation, you will find this axis to reduce dimension and project all your data points onto this vector in order to separate two group of points. Okay. So if you run PCA without labeling, you will get this black line to separate the data there. So if you use LDA, we found this and PC principal component to separate the points. Well, I will first compare LDA with SVM. So this is so LDA is a generative algorithm. So it rotate to find the best separation, use reduced dimension. So SVM is discriminative model. So you can standard SVM is the binary classifiers. Okay, you minimize a function called a hint loss function in order to allow you to find a space between your two classes of data points, and you build a separation line if it's a 2D data, or a plan to separate your data points into two classes. And those classes are pre-trained or labeled. Just before jump to that, I can show you a very simple binary classifiers using lineage algebra, using this example. So this is example from a book called Kernel Machine, the book. They gave the first toy example. Say, it's not an SVM itself, but I'll just show you some of this vector operation and help you to define a very simple classifier and how this works. I show you on the blackboard here. So you have two groups of the data. One is let's see this. So you have some data here. Now you found a center of these data points, and we call it C class here. And then you have another group of data. That's called the C minus, that's center of it. So that's in this 2D space. Now you have a new data point comes in this X sphere. So your job is to assign X with a label either green or red. If red is a C class group, this X is C minus group. Okay, what you can do is to look at, so this is a vector, so this is the data. So how can you set up classifiers? Just use lineage algebra operation to assign this to each one. So the first thing may be this. So you can consider this as a data point. So everything is vector, right? It's vector. You can have a vector or W here. So this vector is not from the origin, but we can move the origin. It's W. W is the difference between C plus minus C minus. Okay, two vectors and difference between that space. Then you found the center of this W here. So this is C. So C is C plus C minus divided by 2. Now you have to compute a dot product. A dot product between this vector, the purple one. So from C, the center to X, and the W itself. Okay. So dot product between these two. So will be C X minus C. Dot product with W. So you know this is a norm X times C times W norm times cosine C. So with this theta is this one, right? So that's a theta. So if theta greater than 90 degrees, then this is a it's 90 and all otherwise it's positive. So that means if you the rate smaller than 90 degrees, you know, this class greater than 90 degrees. So we just use the sine of the cosine theta here to classify. Okay. That's this. C dot W. That's this small example. So at the beginning, this is not a so-called support vector machine, but gradually people adopted this name. I don't know why. In the beginning, it's called support vector network. It can be used to solve both linear and nonlinear problems, work well with many practical problems. That's why it has been widely adopted. So in your classification learner, the MATLAB app, You see their Bayesian method, their LDA, and SVM is a group of method, a class of method. We actually mentioned this in the first lecture when I introduced the timeline of artificial intelligence development. So you see this neuron and the computational neuron is introduced, the concept introduced in 1943. And then that's even earlier than this unique computer been created. Alan Turing's method and the 50. So in 1963, this paper proposed support vector networks. Okay, so that's the beginning of this whole group, whole class of the classification method. So in this paper, the first time they proposed, we need to consider a margin which can be used to separate the class of data points. Well, what is support vector? So support vector means data point, right? The support vector means this one. So what are those points? Those points are the most informative points that can be used to separate the classes. Because this one is the close data point to another class. This one is the close data point to the object class. So those are key points. This is called a support vector. The purpose of this algorithm is to find these support vectors and then give a margin and then give a solution which in the middle of this, and to make this margin as wide as possible. Okay, you can have a lot of different solutions. You have this line that solve the problem. This can solve, but this is the best and the most robust. If you have a new data point that jumps in, it's unlikely to be misclassified. So that's why we need to make this optimal, make it wider as possible, and they found a solution in the middle. Okay, that's the whole point. So this is a 2D examples. If you have a 3D, you have a plan. You have a high D, it's similar combinations to describe, use a parameter to describe this separation plans with maybe hyperplans. Okay, it's fine, no problem. So that's the idea. As I said, you can have many different solutions. This works. Okay, this is fine. This is fine. Which one is the best? You have to have a standard. So the best means you can correctly separate them, and then to keep this separation robust means you can, if this is not measured very accurate, move a little bit because the bias or mistakes in the sampling, but you still be able to separate them and classify any new data points comes into the current categories, right? So we need a better, we need to find this hyperplane to separate them and keep the robust. Okay, so you will see a lot of dot product like this. Okay, so everything at the end reduce into dot product between vectors. So I need to explain to you why this is a solution from the other. Okay, so this tells you the distance between data point to a line. Okay. So, we're done with this simple example for using algebra as a classifier. This is not important. So next example, I want to show you why data points for a line can be defined as the product between. Okay, why you can use this kind of form to be plus some constant. Okay, why this formula works. So this is a Wikipedia page. Explain this the formula, which is this is the distance between data points to a line. Okay. We are not going to use this to explain. But that's a formula, and you can work out by yourself. So I'm going to show you my own example. So at the end, we will see the distance is this. Okay. So the distance is and let's see the distance in this and let's see. Always make this divided by. This is very easy to compute. Okay, so this is your data point, and this contains the information of the line. Is that simple? It's very easy to compute, right? The W and C define the line, and X is the data, and the B is in this form. Very easy to compute. That's why we can very quickly compute a lot of data points to align these things. To find which one is close to this separation line. From this group, which close to this separation line. We need a way to compute very quickly. The very quick way is dot product. Okay. So I will copy this at the bottom here. So that's our goal. We want to achieve this quickly compute data points to a line in order to because the support vector is found the data points which close to this line. This is called the support vector. They're mostly informative data points to help you to separate different classes. So we need to compute all the points to this line here. And then you can trigger the line to compute that again very quickly. So we need the formula to compute this distance. This distance I do right here is times log x minus c. Okay. So the question is, what is W, right? So W is a vector in this 2D example to define the line.
So this line is an example I'm using. So if you know how to write an equation to represent this line, we can see, okay, when x is 0, y is 5. When x is -3, y is 0. So you can define a slope. Slope is x, and this intercept is okay. Just convince yourself. This is a description of line. That's right. When x is 0, y is 5. When y is 0, I think it's -5. Right? When x is -3, when y is 0. Okay. So this in so next week we will discuss iteration. So you have data points. The iteration. This is slope. This is slope. This is intercept. Right. Okay. So now we just rearrange this right in a different way. We times 3 for both sides, and then becomes 5x minus 3y plus 2. Right, this is like a general form to express a line. Same as before. So in this example, you express this line with ax plus by plus c, this. Okay. Same idea here. No problem, right? So we just very general. Now we need two numbers. One is this, one is that. Okay, in the previous slide, this a and b is two constant. So you have a and a b in front of xy, and you have a constant c. So this defines your line, straight line. Here we have three numbers, 5, 15. So I'm going to put these two into a vector W. 5 minus 3. Right? I don't have a name of this vector, but this vector is to tell you the slope information of the line. But this vector is here, so 5, 3, 9, minus 3. This vector point to this here. So this is W. No problem there. So this is my only example. I haven't seen any textbook or online have this kind of explanation, but I just come up with my own way. So this vector gave you the values to define this line here. And if you make this extend to this here, you find this 90 degree of solid weight. This point we call it B. Okay. So V I just gave XY. Okay. So now I'm going to compute to V. Y. I don't know. So I'm going to compute the dot product between these two. Okay, if you place 5 and minus 3 X, Y here put together, you will get this. Okay. That's dot product. And also we know this, right? I've replaced this as a C constant. This relationship should hold because this data point XY is on this curve. So it's on this curve, this expression plus 15 to 0. Follow that. Okay, so that's what we need. Relationship. So move further, so this can be right as a length of that times length of V cosine C between these two. Yeah. So I just replace this plus C. Yeah. So I replace the value of W times V as a magnitude norm, and theta. The theta is 90 degrees. So this is a -1. So this gave you the relationship. What I want to derive here is this distance here. Basically, that's V's norm along this one. I can write this as so this is a 91. This is a 91, so minus this C minus C. Right, this again. This one. Okay, so I replace cosine theta, theta is 90 degrees, as a -1. Okay, so here I replace this with that. And now I replace cosine theta as negative sign here. So with that equation, I can give the length of V is basically that's minus to the plus C. I have a typo in there. So if you move this to another end, and then you divide by norm of W, so norm of V is C divided by this. Okay, so that's what I want. I compute using this expression to compute how long this is. Okay. Okay, we are done with the right side. Okay. So a little bit tricky rearrangement of that, and then get this relationship. This relationship contains C and W, their parameters to define your line. And once you have defined the line and this particular this special point here, the length of this is known. Now we have our data. So our x data is coming in, and this is x1, y1. It's not a very good notation, but you know what I mean here. So I put this as our new data point. Is, for example, located here. Okay, so I will vector. So this is my data point. I want to compute the distance between from this to this line, this curve. So we draw a orthogonal line here, and this at the degree. So that's a d we want to compute. This d is the same as you extend that. It's terrible drawing. Sorry. But that's better. So I extend this particular line to this end. And then this is like a rectangular. So the d we compute this is the same as this one. Okay. So that's what we are trying to get. So this d is a projection of your data point onto this vector, which is from this point to here along this projection minus this. This is d. So the d is our projection of our data point onto this line here. So the projection is x. That's a projection we know from the dot product. So this is the norm of dx, norm of w, and theta. Cosine theta. That's here. Okay. Cosine theta. So the theta is here. Okay? We don't know the value. We don't need to know. And that's projection, which along this part is projection is divided by the norm of this. Okay, so that's an application of our dot product to have that page. So this is along this, I call it D1. This is D1. This is D2. Our D is D1 minus D2. So this is D1. D2 is this. Okay, so D is D1 minus D2 is this part. Minus C. Okay, D1 is here, D2 is there, the difference reduced into this operator. Okay, that's it. So I use this real example just illustrate the distance between this point to here. if you have this line defined by W and C, and you can write the distance for it. Okay, we try to make this as easy as possible to compute in order to achieve the high efficiency of the computation. And also you can do this for many data at the same time. Put all the vectors into a matrix. And the same formula, same dot product, same C to get all the distance between all the data points to this one. Okay. Okay, so you see on this slide, you see a lot of expression. So this is the same as the expression of that. Looks similar, right? So you have to transpose the times your data points plus B divided by the norm. Looks similar. They are exactly the same equation. That this is compute the distance between all the data points to this given line here. The line is defined by A, B, and C. A and B forms this vector, and the C is constant. Okay. So the goal of this, if you look at this expression, is that you try to find the combinations of the arguments or parameters to maximize what's inside here. Well, you found minimize the minimum. So why you have minimum and maximum? Okay. The minimum here is to find points have a minimum distance. For example, from this to the line is minimize this distance compared to all others. So in the center of this equation, you try to first identify those data points, give you the minimum distance from your point to the separation line. And then you try to maximize this is to, after found the minimum data points, those support vectors, you try to maximize their distance, this minimize, so this support vector distance to the line as much as possible. So you found the minimized distance first, and then maximize the margin.
Between the points or
between support vectors, supporter points.
Maximize by the range.
Yeah, maximize the range. So first you randomly select A and B, for example. You randomly select this two, and you can find a lot of different lines, right? One of the combinations satisfies this minimize optimization problem, then you are done. You try to find the parameters to give you the slope. Okay, so you minimize the distance. You found 3 points here. Okay, there's 3 points that support vectors. Then you maximize their separation. Okay. Now, the problem is not so simple because there are some kind of noise. Okay, you may never be able to find a clear separation. Sometimes you have to compromise some of this potential noise. For example, if you point jump into the right margin, if you do a support vector, I don't know how can you put it. There's no way. So another example is if you consider this as the outer layer. And you have to somehow tolerate this impact. Otherwise, your support vector margin is very narrow, but in reality, you want to first remove it and consider this is maybe a better solution. At the end, it's give you the better classification power for new data because this is it's likely to be mislabeled. You have a couple of those. So SVM has this soft margin solution to not only use this absolute optimization solution, but also the penalty of some mistakes involved. So allow you to define a soft margin formula. Okay, so this is just illustrate whether you have hard margin and soft margin solutions. On the example is some outlier or noise or mislabeled data. The soft margin can still survive and give you a reasonable solution. So when you have new class data points coming in, the classification may be not 100 accurate, or but the sensitivity or balanced accuracy and sensitivity so margin solution. Any questions? We are going to jump to kernel. Question? Now I'm going to use this space to define new problems. For example, I have so this 1, 2, 3, 4, 1, 2, 3, 4. So I have 2 data points classified as the ring. So this is position 4, 0. This is a 0, 4. Well, so that's my green class. Well, I have another 2. For example, 1 is in 2 position. Another is in 1, 3. 1, 3. That's my classification problem. I have 4 data points. 2 belongs to red category. 2 belongs to green category. But there's no way I can draw a line to separate them. So this becomes nonlinear problem. If you found a line without clear line, you just cannot draw a line to separate them. Okay, so I think we can come back and see what our solution is. Let's take a 5 minutes break. I clean up a little bit here. So we focus on this small example. Okay, so this is our toy example and not separatable using this linear classifiers. Let me generalize this. So the example of the data point is VI and each one. We have 4 data points, I1 to 4. What we can do in this case is to create a function. This function will take this function. Let's see the function S. Feature mapping function will take this each of this VI and transform VI into a new vector I, y I. Take the same, then you have a ZI. Okay, so this makes the kind of kernel function to map your this 2D space in your high 3D space. And keep the first two the same. We'll replace this VI with the product of this I, yi. Okay, so if you apply this feature mapping function or whatever, or the kernel function, you map your 4 data points in 2D, the 3D. In 3D, and this becomes 1, 3, 3. This becomes 2, 2, 4. So this becomes 4, 0, 0. This is 0, 0, 0, 4, 0. Okay. Let's imagine if you put them onto X, Y, Z, and Z, for these 2 green dots, Z is 0. For this 2 dots is 1 is 3 and another is 4. So you can separate the line between 0 and 3 across the Z axis, make them separable.
Makes sense. So another example. So this example, you have a 2 class. No way you can separate them, but if you found a center and then map those data points, use the information of this particular center, and then put a z-axis on them, right? Which data points close to the center give a high value for its z, then you can make this bent and then become separable in 3D space. So there's no way you can define any plan or linear solution for 2D, but after you do this kernel operation, you map, you expand the dimension of your data to higher dimension, then they become separable linearly. So that's a kernel operation. Another example is this points. These points are in the 4 corners of this -1, -1, 1, 1. Same idea over that. Then you put x, y, x, y. multiplication, then becomes separable. Yeah, question? No? Another example is you use this operation. Okay, this is 1D example that you put them into 2D by take square of the square root of variable and add this to the y-axis, it becomes separable. Okay. So in general, this is function on the kernel function to help you to transform or expand the dimension of the original data, make them separable, linear. It's most commonly used kernel function is called a Gaussian kernel. So in this case, z is the distance between x and z. So z could be in the middle of this one point. So then you compute all your data point to the distance of z. If they are close, then you make it higher in this density mapping. If they're far away, and then you make it a little smaller. So after the operation like that, the point transform just like that. Okay. Yeah.
When do you know when you should apply kernel?
Yeah, that's a good question. So when you can try kernel method, you have PCA, regular PCA, you have kernel PCA, you have SVM standard, you have kernelized SVM. So this is just optional. You can give the try. Then you found, okay, the classifier I create use a kernelized SVM performed really well, and then you can see, okay, the data probably should have this kernelized transformation before it's a separate. It's a good question. The problem, they have some indication that you should or not. I don't know that kind of a selection. Let me show you, play this video. Yeah, always have problem with their videos. So this diagram illustrates the input space. On your left is your original untransformed space, and it's 2D, and you have to use this curve to separate points. Well, after you transform and using this feature mapping function and transform the after kernel, then you can just easily separate them. It's a very simple plan. So in general, and I just illustrate the kernel method, a class of algorithm for pattern recognition, pattern analysis. So support vector machine oftentimes use this kernel to do the transformation first. So here there are kernel functions. Yeah, so also there's a term called the kernel trick. Kernel trick means after you transform your data, it becomes high dimension and the computational, sometimes even simpler compared to your original arrangement. Yeah, so this is called a kernel trick, and there's a technique for you to select correct kernel function. So this kernel function has been deliberately selected, so allow you to apply kernel trick. So the relationship between the data points after the kernelized operation will not become even harder to compute. Well, sometimes it become easier to compute. Okay. So this function you will always see, okay, this is so this is a kernel function, then after the transformation, the relationship between data points. Okay, so dot product. The same as we'll have to expand a lot in order to achieve this. Not going to too much detail of this kernel trick. There are examples here to illustrate after you apply this kernel operation, you get the same results before and after the operation. Okay, after is even simpler, easy to compute. This illustrates the kernel version of PCA. So you know in PCA we need to compute covariance matrix between variables, and look at this variable has already been using the kernel feature mapping function transform, and then you after the transform you do this same thing about this covariance matrix computation, and the points become separatable. Okay, the data looks like this. If you don't do any transformation, you compute the covariance matrix and the PCA gave you unseparatable projection. So you view data from this axis, nothing separatable. But after you apply PCA on transform data, and they're separatable, blue and red dots. We will try this in Thursday computer session and make up synthetic data points, which in the center, there are one class, which another class of data wrap around. So we need to do this kernel transformation, use this Gaussian kernel transform. After this transformation, you run PCA, then they become separatable to one and two. Okay. Any questions? Okay, so we are going to talk about the cell-cell interaction. Let's see. Mathematically, we define heterozygosity using so this parameter, this statistic is being used widely, especially in population genetics. You want to look at a group of animals, human beings, for example. We want to define some genetic makeups, and then we want to define the heterozygosity of the whole population. If you look at by allelic SNP, single nuclear polymorphism. So some points in your genome in the population, they may have either some individuals carry A or some individuals carry C, so that you have this SNPs in the population. And in order to measure the heterozygosity level regarding to this particular SNP, and we use frequency, and if this biallelic SNP have 50-50 between two opposite mutation states, 50A, 50C among the population, then regarding to this particular SNP, the population have reach to this high possible heterozygosity. Since if you pick two individuals out of the population, they look different from each other. The probability is the highest. Well, if you have a distribution of these two alleles, 90%, 10%, the heterozygosity is not as high as 50-50. So how can we measure the heterozygosity level? We use a product of these two values. Okay, if you have this allele frequency 0.5, 0.5, and then your heterozygosity level 0.25, that's the highest you can reach for diallelic. If you have 90%, okay, 0.1, you have 0.09, so it's much lower. Okay, you have a 30%, 70% is also lower. So the idea of this product can be used to measure the heterozygosity. Yeah, so this is the same illustration. So you use 2PQ, okay? P is one allele frequency, Q is opposite allele frequency to measure heterozygosity. Well, because the P plus Q is 1 and squared, so you can expand this equation in this way. So you have a P squared, Q squared, and you can use 1 minus P squared and Q squared to get a 2PQ value. So that's a formula often people use to define the heterozygosity of a group. Again, the heterozygosity definition is you randomly pick two individuals, they look differently. What's the probability they look different? If you translate this concept into interaction, so basically, if you want to pick two potential interaction partners, one should be this form, one another form. What's the largest possibility you pick randomly, they look differently, then they have a potential to communicate. So this idea is used again and again to have the P times Q, so product of two values. So P times Q indicate this off-diagonal position and how big this rectangular area is. Okay, so we are going to use P times Q to measure the potential interaction potential between two genes in two different cell types. As we can see, single cell data differed from a bulk RNA-seq because we have a large number of samples, which each sample is a cell. There are many applications to take advantage of this difference. A lot of stuff that you cannot do with bulk RNA-seq, now we can apply different analysis to utilize the information provided by single cell data. We know we can do cell type identification to find real subtypes of cells. We will discuss differentiation. We follow the trajectory and build pseudo time analysis. we can identify the specific gene program. And in the spatial transcriptome, we have physical location of different cells population. And then I will show you later, we can take advantage of these known positions and to study cell has physical contact with the cell which do not have physical contact, whether they behave differently. Another thing is very important, the cell-cell interaction that you cannot do with traditional single-cell RNA, bulk RNA, but it makes it possible for single-cell data analysis. It's important from this review paper, I copied this diagram, so it gets, you need to study cell-cell communications when you study cell development, tissue homeostasis, especially when you have immune system involved, the cell, different types just interact to each other extensively. There are different subtypes of way of do this cell communications. One is called autocrine. Hierarchin is the form that we will focus on. In this case, you have one cell type produce ligand and another cell type produce receptors. And then if they are established pairs, then this ligand will bind to the receptor to active the downstream signaling pathway to make a change for the target cell type. And the juxta creating is you have to have a physical contact in the creating that you communicate through this bloodstream. And over the years, there are many methods have been developed and published. If you have time, you can follow. There's a link. I collect software tools in different category of data analysis in single cell data. I put it in the Dropbox. It's open to everybody, and I keep updated until several months ago. And there are a lot of packages listed there. Those are the tools that developed for single cell cell communication analysis. The general pipelines look like this. You have first to do the regular thing. You have a QC, you have a cluster, you have cell type annotation because you want to study the communications between different cell types. So you need to define a cell type you want to use as a target and the cell type you want to use as a source to deliver the ligand and this as a receptor. Then you compute average gene expression for all the genes for each cell type. Okay, for each cell type. If you have two cell types, that's the simplest situation. You have two cell types. You compute average expression of all the genes in this population, and then you compute average expression of gene for another cell type. Then you start to look at pairs between ligand and receptor. You get the information of ligand receptor from a dedicated ligand receptor databases. There are data sets people already prepared for a lot of these ligand receptor pairs. You know, okay, this gene and that receptor should communicate. Then you can take those ligand receptor pairs one by one out and compare their expression in these two different cell types. You look at ligand expression in one cell type, and you look at its corresponding receptor gene expression in another cell type. Okay, if you study autocorrecting, you can look at the same cell type where ligand receptor is. You consider the same cell type to have self-communication. But normally we do cross different cell types, one as a donor and another as a receptor. Okay. Or other way around. So this is directional. It's not a one direction, not replaced with another direction. Two cell types. The next, we are going to compute interaction scores for each pairs of this ligand receptor. So what the interaction score is product of this two expression level. As simple as that. So once you compute all the lists of pairs of ligand receptors, then you can see, okay, the top ones with the highest interaction score can be fished out, and then the report and the further study. Make sense? Okay. Just like that, it's very easy. But very easy, why there's so many different yeah, so some of them, for example, like this is just a collection of either customized ligand receptor database. So itself, I wouldn't call it like as an algorithm, because there's no algorithm developed at all. It's just customized. They collect a lot of ligand receptor and put it into a comprehensive database. It's also a contribution to the science, but it definitely should not be a tool. A lot of this provide a simple interface writing in R, so people can easily compute the product between two genes. Again, it's not an algorithm. It's just put together some pipelines. Some of these are useful because they are not only consider one pairs of the ligand receptor, they probably consider three or a set of ligand receptor. They know, okay, they work together all the time. So instead of compute individual interaction scores for every pairs of these ligand receptor pairs, they probably compute a ligand receptor set. And then, okay, another way, or ligand receptor interaction involved in the same pathway. They make a pathway level of this score. Again, this is not a very creative. It's engineering. It's some kind of software engineering. Or they build not only consider two cell types, they will consider 5 cell types and then compute pairwise different communications and build make a graph. Okay, but at the end, it's also still the product between ligand receptor gene expression. No matter how fancy you put it on the top, there's no advice in terms of algorithm. Okay. So you need to know those facts. So when you apply, say, I use niche net, I found some of this, but not in another pipeline. I found a different. Which one should I use? Well, you have to know a little bit more about the underlying principle why they published doesn't mean it's a reasonable advice. Okay. So this is an example of a ligand receptor database. There are many of them. So this is one of these. It's called a cell linker. Also, sure you can see. You can download some of the text files, and in these text files, you see these columns. So this is the donor, this ligand gene, right? What I can see is ADN2 and the secreted. Okay, and then here's ACVR1C membrane. So this is the receptor ID. Okay, some relationship memory and the statement protein, different ways to communicate. So this is just a long list of the pairs, gene pairs. So once you have that, and then you start to look at your expression level of your cell type A and B. So this is like gene 1, receptor gene 1. So let's show you the average expression of a cell. Okay, all the cells, on average, its level is 3. Well, for cell B, the average of this ligand is 0, not expressed. Well, for the receptor, it's opposite. Cell type A have no expression, or cell type B have some expression. So how can we compute interaction score for from cell type A to cell type B regarding to ligand 1 to receptor 1? It's 21. Okay. Is this opposite? It's a direction opposite. So the cell type B become a donor to produce ligand 1, and then cell type B A becomes the receptor producer. Okay. Also, the score is 21. So this analysis is directional, so you cannot replace cell type B. The direction from A to B, B to A is not the same, not symmetric. So if you have, in this case, we just don't consider them. Okay, the score is 0. We will play this example data search data. So this is, I just randomly select the data set from a mouse skin, wounded tissue. If you gave this session number ID, the program will automatically download this matrix file. Then we will do this regular thing, dimension reduction, then cluster cells, annotate. Okay. So we will try this with this real data. Anyone knows this stellate cells? No. The first time I say, okay, stellate cell, I saw this in the central nervous system. I was worried about this misannotation of cell type. But later I Googled and searched, and in the skin, you also have a stellate cell. So it's reasonable to have that. It's probably migrated from the liver. The liver has stellate cell, and the pancreas has stellate cell. So someone suggests those cells in the under the skin, probably because of the immune system work, and some of the liver or pancreas stellate cell move to there. I have no idea, but it sounds like reasonable. It's not maybe not a mislabeling of the cell type. Yeah, so this is a support I found from this review paper. Stellate cell are resident lipid storing cell that pancreas and liver that transdifferentiate to myofibroblastic states in the contact tissue of tissue injury. So it somehow makes sense. We have a mouse wounded injured tissue, and this stellate cell is there, so it's fine. So in the database, I just randomly pick. Don't remember. Maybe I didn't randomly pick, but I know the signal is there. So I just pick this pair. And these pairs, there are explanations of these pairs. One is receptor, and then one is a ligand. So there are strong evidence support the interaction between the two. Okay. Then I just compute a score between. There are three different cell types, so I compute the score for each of them, and for both direction. At the end, I found that the score is higher for this particular ligand receptor pair, and this highest is from the endothelium cell to stellate cell. So those linker, the thicker, the higher the score is. Okay. And then they are directional, so this indicates this will produce ligand, will produce a receptor. So if you have more than two cell type, you can create some kind of this map. Okay, you put a score in there. Then I come back to look at expression of these two genes by just using this heat map profiles and expression level in endothelial cell for this ligand gene. Okay, you know how to look at this. And then correspondingly, the receptor gene expression in what I call the target cell type, stellate cell. So that's a profile. And if you look at this bar plot, this is not a marker genes for endothelium cell, but you can feel the expression level is increased compared to other cell types. Well, it's a receptor gene, so highly expressed in stellate cell. Okay. By the way, so here we use average expression of gene. So there are some problems. So in this case, it's fine because almost all the gene cells have upregulated this gene expression, but sometimes the upregulation, the average maybe not the best way to measure the upregulation particular gene on average, especially if you have a lot of zeros in the population for that particular gene, and you upregulate just a few cells, the average will not change a lot once you average across the population, because the baseline expression across all the cells are low, so there are a lot of zeros in there. Yeah, so we have to consider this too. Maybe we should develop a new method to not only use the average expression, but use additional information or distribution, something within the population of a particular cell type to build a better score. Okay. So the recent developments in this the cell-cell communication package we developed in our lab. Yunjian play a major role in this development. So this is a pipeline we created. Yunjian, maybe you can explain this to us, or you want me to do it? You still have like 15 minutes to explain your pipeline a little bit. Yeah.
I hate it here.
Yeah, you can come here because you develop this. You know the best.
I don't speak, but I try to speak loud. Okay. Yeah. Go ahead. You can use. It's fine. The basic motivation behind developing this such a tool is that we want to integrate the neural network, the modern machine learning framework into the inference. So as you can see, there are two neural networks, but the big picture underlying this framework is that we want to project two SEGRN, the gene regulatory network of cell type A and B into a unified low-dimensional space. So here, each dot represents a gene from two different cell types. So here, the purple dot is a gene from the purple cell type, and this blue one is a gene from cell type B. So once we have this, once we have the low dimension representations of genes from two cell types, like this, we actually project into the 3D space. Here, I guess I show the 2D vector. So each gene is a 2D vector. It has the coordination of itself. So we can calculate the distance between each gene from two different groups. Here we just use the Euclidean distance because we believe it's an orthogonal space. So we calculate the distance between each pair of genes in the low dimensional space, and then we obtain a distribution of a distance of each pair. So if we have, for example, if we have a 10 pair of genes, we will end up with a 10 by 10, 100 pair of distance here. And it turns out the highly interactive gene pair located in the left tail of this distribution. So we will have a test, a statistical test to get the significant ligand receptor pair located in the left tail of this distribution. That's the reason why we query the prior knowledge database, the ligand receptor database from here to extract the ligand receptor, the significant ligand receptor pairs which were located in the left tail of this distribution, and that's the thing that we want to infer. And the key question, the key technical question is that how we obtain this unified, how to obtain the low dimension representations of genes from two different cell types. So here, I think the key motivation is that suppose we have a network A and we have a network B. We want to project them into a low dimensional space like this. This is A. This is B. Suppose we project them into the low dimensional space so that the matching could be two. Because we are commenting, we like the 2D or 3D space, we can visualize that. So here the network is the, suppose we have 100 genes, so it's a 100 by 100 space. This gene is a 100 dimension vector, so it's really hard to visualize them. So how to project them to the 2D space? Intuitively, or the most straightforward way that you can think about it is use for example, PCA that we learned in the past. Of course, we can use PCA to project this field to the 2D space, but it turns out it doesn't work because we ignore the relationship. We call it correspondence between these two networks. So we still need another constraint. So this is we call it correspondence, but it turns out, it's just that how, suppose you have the gene 1 from here, and you have gene 2 from cell type B. The element here is just indicating how strong they interact with each other. So if you have a ligand in the context of cell communication, if you have a ligand here, you have a receptor here. So this element in the correspondence matrix indicate how strong they interact with each other. So not only you have the information from the string network, you also have their interaction, have their how strong their relationships are. So by mapping, by projecting this records the by projecting this whole matrix to here, we will have not only have their local similarity but global similarity, meaning that we maintain the similarity between the cell type A and B, and we also maintain the similarity between cell type A and B. So we have 3 similarities. We have similarity in A, we have similarity in B, and we have similarity between A and B. So this is the comprehensive result considering these 3 factors. Of course, lastly, before we have this neural network, we use the PC regression, principal component regression to build our gene repertoire network. This is work developed by my senior, not by me. And this is a really great method to have the network, gene network, from the single cell RNC data. And if you're interested, you can try that. It's called IC24NET. This work is called IC24ICT. And the first one is called IC24NET. And if you're interested, you can search online. And that one is also good. I think that's the end of my presentation. Do you have any questions?
The other S will be interaction between the
Yeah. Yeah, that's the motivation that we get. We can use 2 PCA. We can use 2 PCA. Dimension then reduce the dimension from 122 122. But in this way, you don't have the row star don't know the relationship between them. You have actually this now. Oh yeah, you can search online, and I think they will lead you to my to our lab's GitHub.
You also think why we take the first portion of the distribution of the distances as the same between thirds?
You mean why the left tail, not the right tail?
Yeah.
That's a good question. It's in the we call it, the algorithm actually called manifold alignment. Actually, if you have a really strong, really larger value in this relationship, the distance in the low dimensional space would be so close.
The 2 genes close together, one is from cell type A, another from cell type B. These 2 genes close together means potentially stronger combination because they have information in the corresponding matrix group. Tail that's potentially stronger. Yeah. But even you have very weak corresponding because of the push of the 2 networks, maybe push them together. So you can have test this to compare with a lot of those other published, our is the best for the time.
I think you just kind of ask the correspond between the A and B. I think receptor and ligand. No, we use all the gene. Yes, it's a good question. We use all the gene pairs. And here only until here we query the ligand receptor database. So that thing is just all the pairs of gene.
But one more say correct. If you only want to evaluate non-ligand receptor, you can put only values in the S here between non-ligand receptor combinations. But in this case, you can put everything to discover some of the ligand receptor which not in the database.
That's good.
Other no questions?
I have a question. Whether this is only very significant of set of
What do you mean by sub?
therefore the subtype is the T cell, it's only
Oh,
significant in the
no, we didn't implement the feature selection. We use all the genes. And if you want to speed up the computation, you can use usually in the field of single cell, people use so-called highly variable gene. Because we will have more than 10K genes, and most of the genes are now quite informative for your downstream analysis. So usually people just select 3K or 5K genes for downstream. So here, I assume you are asking whether this matrix is manually selected subset matrix. No, we didn't do any filter on this. I think that you focus on the normal the cell type, no other kind of treatment. But you might just have the treatment want to just determine between the difference. Oh, check this. Yeah, actually we have another diagram.
Yeah, so your question is good. You want to compare the change of interaction. So what you can do is you can independently apply twice, once on your condition 1 and condition 2. This is one way, but that's not the best way. Let me see my other. This is just separated illustration. Yeah, so we do have the comparison between two samples.
So this one is the one that I drew here. This is called the joint similarity matrix. So we have WA and WB representing two networks from subtype A and B. So here we will have two single sample joint similarity matrix located in the diagonal. Right? So you have condition A, condition 1 and condition 2. So you will have two single sample joint similarity. And here we just put the identical matrix to enforce the correspondence. So by doing this, we avoid the pitfall that people usually run their algorithms twice for different conditions. Here we will have the constraint to enforce the correspondence between these two conditions, and it will end up with at least by my test, it will give me positive results.
This setup will help you to find the differential interaction pairs. And how interaction become stronger in one condition or become weaker in one condition, something like that. Contrasts.
So if you want to test whether treatment reduce the activation of some receptor cell, and just complete each like A in the control and HB in the treatment cell.
Yeah, we don't do computation twice. We don't trade independently. We put them together jointly, internally jointly. conditions.
And then see that interaction decreasing.
This is much sensitive. But this one, you have to use cluster computing because it takes much memory, large memory. You have to put all the values in there.
So normally we use from first point or.
Yeah. Grace order should be fine. Yeah.
That we keep the longer time to finish this stuff.
Yeah. Because just as I introduced, I need two neural network to solve this. So therefore, I need 4 neural network to solve this. So it will eat your memory if you use a regular PC.
Still, do we need to complete all the teams? And then,
We can just use the highly variable genes. 3,000, based on my experience right now. It wouldn't, even if you use 5K genes, it wouldn't significantly change your downstream result.
The high variable genes between the big cell.
No, highly variable genes. You can, you can Google what is highly variable genes. Usually, in the field of single cell, people use packaging like Surat to select the highly variable gene.
so the highly variable genes is providing.
No, highly variable genes is just genes across all the single cells. Their variability, their variation really high.
We will discuss highly variable gene next time, next lecture. After that's very important. Yeah, in the package we have 2 different ways to do a highly variable gene. So don't use the Harry short. Actually, we have the best way. The other people test our method is the best. Yeah. Yeah, so highly variable gene means in a homogeneous population of cells. And that some genes express, for example, similar level across all the cells. Those are not highly variable genes. You know, some other genes express level is very different from other cells. Those are the most important genes in single cell. So I have a whole lecture on highly variable gene and their biological significance. Okay. So in this case, we want to reduce the computational cost. So we select highly variable gene to make 10,000 genes matrix to 5,000 genes. Because those housekeeping genes in this region are the same are not very important. Let's see. Oh, so that's the further development. So we have also consider whether there are physical contact between different cell types. Take that information into account. To, for example, this is like a solid tissue, and then you have interface with the immune system and immune cells in the mesenchymal. And so if you have physical contact, those cells will behave slightly different from a cell which is inside the solid tumor, which not been exposed to the immune attack. So we try to detect this slightly different. Thank you, Yuja. That's our other slide. Yeah.
Is there a way possible like we can calculate the end of time communication between different cells?
Interpret.
Like autoplay or directly content?
So interpret is you're negative carried by the bloodstream, so you have a long distance transfer of this information. So probably if you have a single cell data, one from one location and then from the targeted tissue, that will be really cool. Detect whether there's some interpret relationship. If you have a real good control, okay, I know this information, and then you can block some information transfer.
So we combine the two data sets from the single cell. From one.
I don't know anyone has already done this. I have never heard of anyone studying this interference single cell communications. That's really a cool idea. Good suggestion. Yeah, so that's all. Okay.
I wanted to ask you about the homework.
The homework, yes.
What do you? Like,
Yes, I believe if you create a classifiers, we have to use some of them. Method will be fine if you use others. Acceptable. But I don't think that Excel itself. Because there's a treatment of large matrix with a response variable to classify patients. By looking at the text matrix. There's like how we yeah, so the project will be at the end have a presentation. Yeah. A presentation, I don't know how long it will be, so be 10 minutes. And then 5 minutes so from previous semesters, other students present their students after they learn all this technology, how I apply and the hypothesis and which study what they want and generally describe what they learn. The data sets you can have them from a geodatabase. You see, just like this, the mouse data. All you need is the ID. Let me show you how to do this. All you need is this.
Is there like a particular location to get those IDs or like based on?
This ID you search geodatabase and maybe you search 10x genomics, single cell, and also disease name you want to study. You will get a lot of this.